Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Mary Gehring from MIT on the show. Mary, please let me briefly introduce you to our audience. You received your PhD in 2005 from the University of California in Berkeley, where you worked with Robert Fisher. You then did a postdoc with Stephen Henikoff at the Fred Hutchinson Cancer Research Center. You then started to lead a laboratory focused on epigenetics at the Whitehead Institute in 2010, and you are now an associate professor at the biology department at MIT. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
I don't know if I remember exactly when I became interested in biology, but I grew up in a very rural area with lakes, woods, and so I spent a lot of time outside. My family had a large garden, so I spent a lot of time doing that, weeding the garden. And my dad was a high school biology teacher. So there was a lot of, biology in the air in my upbringing. So I really liked spending time in the natural world, and then also had this influence at home. And then when I went off to college, I actually thought I wanted to major in English and be a writer. So I think I am a writer in some ways because I, write grants, write papers, but not the sort of writer that I envisioned. But then when I was at college, I really loved genetics and I loved plant biology. And so I started working in a plant biology lab, and that's what got me started in being interested in doing research long term.
So did you have an academic background in family, or did you know what you were into when you started doing biology, like following the academic path, or was this something that you learned along the way?
No, I didn't have an academic background in my family. Although my older sister, who's 4 years older than me, she went to get a PhD in biochemistry. So I knew somewhat of her experience. Although even then, I remember I would ask her when she was maybe a year into her PhD, like, "Oh, when are you finishing?" I didn't really understand what a PhD was. So I didn't have any experience of any parents or other relatives being in academia.
So coming to your science that centers around epigenetic mechanisms of gene regulation in plants, I must confess that epigenetics in plants particularly has not been featured very frequently on this podcast yet. So before we dive into your work specifically, maybe we can take a step back and ask the question whether there are any major specific differences when it comes to epigenetic mechanisms in plants versus the mammalian system.
There are some differences, but it's a little surprising you haven't had a lot of plant epigenetics on your podcast, because
I might be biased.
Yeah, really, that's where epigenetics started. Some of the work in maize or corn in the 1920s, some of the epigenetic phenomenon like paramutation were discovered then, or in the 1920s, 1940s. So there are some differences, but mostly similarities. I mean, in terms of chromatin and histone modifications, all those same sort of modifications are present in plants and animals. I work on DNA methylation, and in plants, the DNA methylation system is a bit more elaborate than you see in mammals, for example. So there's DNA methylation, more context. And the mechanisms of demethylation are somewhat different, but overall there's a lot of similarities. I would say in plants there's much better evidence for things like inheritance of epigenetic states across generations, and there's more examples of epialleles actually being important, perhaps in an evolutionary context, than there is for animal systems.
Is it because it's easier to study, or because there is just more of the pathway?
Oh, you mean like in terms of like epialleles, is it easier to find them in plants, or is it?
Yeah, or if it, because of the model organism, it's easier to handle, that you can do the experiments easier because of generation times and things like that?
Yeah, I mean, the generation times are not necessarily quick. We mostly work on Arabidopsis, so that is fairly quick for a plant, but certainly not like working in yeast or C. elegans or flies. I think part of why some of these phenomena are more likely to be important for plants because of the way plants grow and develop and respond to their environment. Plants undergo continuous development throughout their life, which is quite different from an animal system where there's growth, but not what we would call development.
So let's dive into your work. I want to start in 2009. Then your first science publication came out. There you profiled Arabidopsis DNA methylation genome-wide in the embryo and the endosperm. What did you learn from the genome-wide data there?
I like how you call it my first science publication. It's my only science publication.
But maybe when people listen to this in 5 years, there will be more.
So we were interested so we already knew going into that the endosperm was where genes were imprinted in plants, meaning genes are being expressed differently depending on they're inherited through the male or the female. And but really, there had only been a couple of genes that had been looked at. And that was part of what I did as a graduate student, look at this one particular gene, Medea. So I was really interested in Steve Henikoff's lab, learn genomic approaches, as that's what I wanted to do for my postdoc. And that was just at the time when like high throughput sequencing was becoming a thing. So when I first came to the lab, they were still doing methylation pull down and then hybridizing on arrays. And then for this experiment where I looked genome wide in the embryo and endosperm, I did like a methyl IP and then sequencing, which was I think the first time in the lab we'd done it was called Celexa then, Illumina now, high throughput sequencing. So what we found is that there were these genome-wide differences in DNA methylation between the embryo and the endosperm. I say genome-wide, but still at discrete sites, that basically the endosperm was hypomethylated, but hypomethylated particularly at short fragments of transposable elements. And this was quite exciting because some of these demethylated regions were associated with imprinted genes. And we had the demethylase mutant DME, and we could see that these regions became then hypermethylated in the DME mutant. So we knew that these were genetically controlled by DME. And so we were able to find that for imprinted genes, their regulation was now tied to the epigenetic status of a nearby TE. So that was very exciting to uncover this more global mechanism of imprinting and identify many more imprinted genes than had been known before.
So this was like, you said you did an IP for that. So it was not like looking, I mean, it's still genome-wide, but it was like an enrichment first and then looking what you enriched for, right?
Right. It was like an IP-seq. So it was a methylated, it was the methylated DNA antibody. Yeah.
So next, you followed up on this with a paper published in eLife, and you looked at gene imprinting then in Arabidopsis. Can you talk about imprinting in Arabidopsis and what you found specifically in this study then?
Okay, I'll have to remember back to that paper. Yeah, that was I guess one of the first real papers to come out of my lab. So in this paper in Steve's lab, I had looked at two different accessions of Arabidopsis or strains. So Arabidopsis is distributed around the world, and there's lots of natural genetic and epigenetic variation. And what I had found in my postdoc is that I noticed that some of the regions that were demethylated were distinct between the strains. And I thought, maybe that might be connected. Perhaps there's variation in imprinting in Arabidopsis because of this, and maybe that's connected to seed phenotypes. Because we knew that at least some imprinted genes were important in controlling seed development. So in the eLife paper, we set up this system where we took 3 different strains of Arabidopsis, and we did all these all possible reciprocal crosses among them. And so we were able to identify genome-wide imprinted genes using RNA-seq, and then we were able to find imprinted genes that were just imprinted in one strain but not another. And we then were able to connect this to variation in natural variation in DNA methylation in those strains. So this was really interesting to me because it suggested that natural epigenetic variation that exists can have a potential gene expression, and then in a later paper in 2018 in PLOS Genetics, we showed a phenotypic consequence for the seed.
So it's like an influence from the outside has an effect on the epigenetics, and it's not like from the inside from the plant.
Well, I would say we don't know. We don't know that the difference in DNA methylation that exists is because of an environmental influence. We know that variation is there. We don't know how it got there.
So we now talked mostly about DNA methylation. However, at least in mammals, there is also 5-HMC, so hydroxymethylcytosine. You set out to test whether there is 5-HMC in Arabidopsis, or maybe specifically Arabidopsis or plants in general.
Yeah.
Did you find any?
Well, that was sort of a saga. So it was. This also comes back to an antibody. I don't remember if we bought the antibody from where we bought it, but anyway. So this was the last experiment I did before I finished my postdoc. I did an IP with a 5-hydroxymethylcytosine antibody,
which would be logical if you did the methyl experiment,
right? And did like by dot plot, you could detect something in Arabidopsis, and I also did some other related species, Arabidopsis lyrata, a couple others that I had lying around, and did the IP and sequencing. Or maybe I hybridized it to some old chips we had, but it looked like there was HMC. But then, my poor first graduate student, Rob Erdman, I got convinced him to join the lab based on this project. Like, we're going to figure out what HMC in Arabidopsis does. But it turned out that any other method we used besides the IP, we couldn't detect it. So eventually, with mass spec, we couldn't detect HMC in Arabidopsis. We got some corn DNA, couldn't detect it there. And so we concluded, if it is there, it's at a level below we could detect, what we could detect by mass spec. So I think in Arabidopsis and in other plants, it's just methylcytosine and then direct removal of methylcytosine by the 5-methylcytosine DNA glycosylases.
Yeah, that would have been my follow-up question, because would that mean, but now you kind of answered that there is no 5-HMC, or it's just like so transient down the road for 5-FC or 5-carboxycytosine and they might be there and have an effect, or is it just 5-MC or nothing?
I mean, I can't, of course, exclude that there is some of these bases in DNA, but there is no, for example, homolog to the TET enzymes that has been found.
Then.
And so I think probably not, but there could be a particular tissue or stage development or something that hasn't been annotated correctly yet in plant genomes that might have this activity, but so far it doesn't look like it.
So next you investigated imprinting further, and that was probably then in your own lab by looking at imprinting in Arabidopsis lyrata. Why did you need to look at this species? So why did you switch, and what did you find then?
Yeah, so Arabidopsis lyrata is a close relative of Arabidopsis thaliana. Well, it's not that close. It's about 10 to 15 million years diverged, but it's the closest. And unlike Arabidopsis is mainly self-fertilizing, but Arabidopsis thaliana, but Arabidopsis lyrata is an obligate outcrosser, meaning it can't pollinate itself successfully. It has to be crossed with another. So after I first started my lab, I got invited to join this group called Testing Theories of Genomic Imprinting, which meant at NESCENT, which was this evolutionary, I forget what NESCENT stands for, but Evolutionary Synthesis Center that was at Duke. And it brought together people working on imprinting for multiple systems at the molecular level, and also evolutionary theorists to think about what does evolutionary theory say about imprinting? Why did it evolve? How is it maintained? And then what does molecular biology say? And can we bridge that gap? And this was very instrumental in my thinking about imprinting. And this is going to be kind of a long answer. One of the theories about why imprinting was selected for is because of parental conflict between maternal and paternal genomes in offspring tissues, and particularly in offspring tissues that influence resource transfer from the mother. So in plants, that tissue is the endosperm, and in mammals, that tissue is the placenta. And basically, the degree of parental conflict should be greater in Arabidopsis lyrata than in Arabidopsis thaliana.
Because it's not the same.
Right. And so I wanted to, and there was a particular prediction that paternally expressed imprinted genes should be expressed more highly when you have greater parental conflict. And so that's why I wanted to compare imprinting in thaliana and lyrata. And what we found was that indeed, many of the imprinted genes were the same, but the paternally expressed imprinted genes were expressed more highly, had a higher dosage in Arabidopsis lyrata endosperm than thaliana. That's one of my favorite papers, actually.
I will make sure to link it in the show notes. Another question when it comes to imprinting and inheritance is how stable is the information over the generations? And you also looked at that. Can you tell us more about what you find there?
What paper are you referring to?
That's a good question. It's like at a DNA methylation sensing circuit.
Oh, well, that's not about imprinting.
No.
So that is, this was something that I actually first found when I was a postdoc, but didn't publish until I had my own lab, and a new postdoc came to my lab, Ben Williams, who really worked on this a lot and figured out a lot of this. And so that's centered on one of the 5-methylcytosine DNA glycosylases, ROS1, which is primarily active in vegetative tissues. So this is an enzyme that removes methylcytosine from the DNA, leading to its replacement with cytosine, which is how demethylation occurs in plants. And so we found that ROS1 is itself regulated by DNA methylation. I should say that actually Marjorie Matsky and others had already found that ROS1 expression was affected in DNA methylation mutants. So basically, and Zhenkeng Zhu found very similar things as well to what we did and had a paper in the same year. So the ROS1 expression is controlled by methylation, but in an opposite way to typical genes in the genome. So methylation in the ROS1 5' region at a T fragment actually promotes ROS1 expression, and loss of methylation or demethylation actually leads to decreased ROS1 expression. And ROS1, we showed, is demethylating its own 5' region. So it sets up this very nice regulatory circuit where when...
Kind of a feedback loop.
Exactly. Where we think that ROS1 expression levels is responsive to overall methylation activity in the genome through the action at its 5' region. So when methylation activity increases, you have increased demethylation activity. And so our idea was that this might be important for maintaining epigenetic homeostasis. So what you're referring to in terms of stability is that we found we could basically uncouple this circuit between methylation and ROS1 expression. And what we found is that there was a degradation there was a loss of epigenetic DNA methylation information from one generation to the next. So it got progressively worse.
I have a question in mind, but I can't express it.
Okay.
So I'll just move on. You also mentioned it earlier a little bit that there are different tissue types, and that methylation might be different in different tissue types. So this is a paper that was published last year, so 2022, if I'm not mistaken.
Yeah.
And you looked at tissue-specific methylation states and its impact. What did you find there?
Yeah, so we were looking at what we were able to do. This was Ben Williams' last project in the lab before he left to start his own lab at Berkeley. Is there's 4 DNA demethylases in Arabidopsis, but one of them, DME, is essential in reproductive tissues because it has this. It's involved in establishing imprinting. And so we hadn't been able to look at a quadruple mutant before to basically look at a plant where there was no demethylase activity. So what Ben did is he just complemented a DME mutant in the reproductive tissue only. So he expressed DME under a reproductive specific promoter. And so then we had plants that we can now make plants that were null for demethylase activity in somatic or vegetative tissues. And what we found is we identified more demethylated regions than we had seen previously. And also what we found is that many of these regions that are demethylated can vary in methylation state among tissues. And so whereas previously the field would think about, okay, a tissue-specific methylation pattern is due to differential methylation activity. What this work showed is that part of the reason there's differential methylation patterns is due to differential demethylation activity in different tissues.
So the other end of the spectrum.
That's right. I mean, it's maybe obvious now saying it later, but didn't seem so at the time.
It depends on your viewpoint, right?
Yeah.
So to finish off the trip through your scientific career, I want to talk about the method CUT&RUN.
Oh, okay.
Being influenced by Stephen Hendikoff, it seems obvious that you are also interested or influenced by his methods. What did you do to adapt CUT&RUN to plants?
Yeah, we didn't have to do too much adaptation actually. So I may be forgetting some details, and I'm sure Xiaoyu would not appreciate me forgetting those because she's the one who has adapted it. But it didn't require too much change from the protocol that the Hendikoff lab had already put together for Drosophila cells, mammalian cells. We do it on nuclei, not whole cells.
That's probably
that would be the main difference, because plant cells have cell walls, and so if you want to get out a cell, you can do what's called protoplasting, where you digest away the cell walls. But for us, it's just easier to isolate nuclei and do it in nuclei.
So that's basically the main step that differs.
Yeah,
the sample preparation.
Yeah,
and then it's also useful for all tissues in plants that you can use.
Right.
So how does the CUT&RUN, or how did it influence your work? Is it like just another method that is now sexy, or is it like, does it have really big advantages to other methods like cut and take or chip?
Yeah, I think that it has influenced our work because a lot of our work, as we've talked about, is in this tissue called the endosperm in Arabidopsis. And the endosperm is, a tissue with an eye inside a seed. And I'm sure you or most of your listeners have not seen an Arabidopsis seed, but they're very, very tiny. And so, I was always wanting to do chip. And actually, I had first tried intact to isolate those nuclei specifically. That didn't work very well for me. But with CUT&RUN, because the input that you need is so much less than for chip, it actually now opened up our ability to look at chromatin modifications in the endosperm and at allele-specific chromatin modifications in the endosperm that we can now connect to things like imprinting and other gene expression programs. So yeah, it's been really great to be able to do that.
I can imagine that. So, okay, now really the last question on your science. What is it that you're doing right now, and what will you be focusing on in the next 5 years?
Oh, next 5 years? I don't know.
Or for the next grant, what you can like.
One thing I'm excited. I'll just tell you a few things, or one thing. So we found. You didn't ask about this paper, so I'll mention it. We were interested in looking at imprinting at the single cell level in the Arabidopsis endosperm, because based on our bulk sort of RNA-seq, we would always find that particularly for the paternally expressed imprinted genes, that they would never look completely monoallelic. And so we wondered whether that was due to, both alleles are being expressed just at different levels, or whether there was variation in imprinting among endosperm cells or endosperm nuclei. And so former graduate student Colette Picard looked at this with single nuclei RNA-seq and identified that there's imprinting variation within the Arabidopsis endosperm, and particularly we find that there is stronger imprinting of paternally expressed imprinted genes in the callosal endosperm, which is a region that sort of interfaces with maternal tissue. So that suggests that there must also be epigenetic heterogeneity within the endosperm that we've been blind to before. And so what we're doing now is trying to understand that. And so trying to understand whether within the endosperm there's different epigenetic states that are connected to this variation in imprinting. And so now we're, doing things like CUT&RUN on even smaller populations.
Or single cell?
We haven't tried the single cell CUT&RUN. I think we might try that this summer, I guess. There's a student coming to try that. Understanding what are the sort of epigenetic dynamics that are occurring after fertilization in the seed that explain these differences in imprinting.
Sounds very interesting, and we will keep an eye out for that. So now science is finished, but I have rather two more general questions. So the first one, did you at one point of your career face a situation that you have reached a dead end or hit a wall where you did not know how to proceed to unravel the questions you wanted to answer?
Oh, many times.
But is it like a micro bump, or is it like
a project that you just stopped because it didn't work?
So both ways is fine.
Yeah, I mean, I think in all projects there's lots of bumps, as you know. I would say the HMC project surely hit a wall.
But no result is also a result, right?
That's right. I mean, it's harder to get no results published, but I mean, negative results, I should say. They were great results, very beautifully done. But I mean.
How did you then cope with that? So have you been trying to look even harder to find something, or did you say at one point, well, the data is conclusive enough, so we stop here and we know that it's not there?
Yeah, basically that's what we said. And then we said, and then there's nothing to publish. But after some reflection, probably after a year, we were like, well, maybe we should publish that and tell people that there's actually not HMC in Arabidopsis. So it turned into something positive. And I would say that was really actually led by the graduate student who, obviously wanted to get something out of the nice work he had done. Yeah.
So in the last almost 30 minutes, we have taken a journey through your scientific career. And this is why I did not mention all your papers, because I wanted to give you some room here. But can you maybe give a short summary about your most important findings or something that we might have missed? But
I did already. I spoiled your question. I'm sorry.
That's okay.
Most important findings. Yeah, that's tough.
Or something that you like most. Maybe.
Yeah, I mean, I guess I really like the ROS1 regulation by methylation. We coined that the epigenetic rheostat. I really love that work, and I think more and more I'm interested in evolutionary biology, and so.
So you're thinking of including more plants into the panel and looking at.
Yeah. We've done some work on water lily that hopefully we'll have a paper together soon. So water lily is really interesting because it's a very early flowering plant. It's an early diverging angiosperm, and endosperm. The genetics of endosperm are somewhat different in water lily. And so we've looked for imprinting there and have some interesting results. So I'm excited about that. But it also, leaves open questions that would maybe be answered by looking at additional species. So, yeah.
Yeah. So thank you, Mary, for your time and for being on the show.
All right. Thank you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.